Hi, good morning. My name is Natalia Elizalde, Business Development Director at VIVEbiotech. We are a company fully specialized on lentiviral vectors, so I'll try today, explaining you a little bit more about what we are doing and what our expertise is. Okay? So we are a GMP CDMO, a GMP contract development and manufacturing organization fully focused on lentiviral vectors. But apart of offering this service, what is very important is that we are continuously working on the optimization of production process, both from the upstream and downstream point of view, putting great effort in increasing titers and of course in enhancing purity as well. That is also very important. Moreover, our aim is enhancing the properties of the viral vector from different point of view, making it regulatory acceptable, but also, safer profile is one of the key issues we are working on. As a company, we are working on the development and manufacture of lentiviral vectors from scratch, even from cloning, R&D manufacture, and so on, to GMP manufacture of lentiviral vectors. So at the moment, we are manufacturing batches for phase 1-2 clinical trials, but we are also involved in an expansion plan to give answer up to commercial stages. So later on, I try to explain you more about this expansion plan and where we are going in the short term. Our aim is adapting process to every lentiviral vector and also developmental phase. And for that, we are optimizing this production, considering both titer and purity, as mentioned before. Also, we think it's very important starting from very early stages, like from scratch, to avoid potential regulatory hurdles later on. And also, scalability and cost-effectiveness issues are being had into account from the very early beginning. We are an international company, which means that we are working for different companies globally. Here, in this world map, you can see marked in green the different countries where our customers are based. So basically, we are now working for 30 worldwide projects. 50% of them are based in the US, so these customers, they are based in the US. Also, we are working for different companies based in different countries in Europe, such as Norway, Italy, Switzerland, UK, as well a couple of customers. One Asian company based in South Korea, and also our lentiviral vectors are being used in Australia. So basically, as a summary, what it means is that we are working in compliance with the main regulations, compliance with EMA and FDA regulations, indeed. A key issue, or something that we consider fundamental, is being able to offer slots availability in a timely manner. As you all know, this is kind of a hurdle while developing a therapeutic that encompasses the use of a viral vector. So that's why for us, ensuring slots availability in a timely manner is one of our aims. At the moment, we are able to manufacture up to 20 batches in reactor per year, but in 1 year time, we'll be manufacturing 40 batches in reactor per year, and in 2 years, it will be 60 batches in reactor per year. What it means is that we will have the capacity to manufacture more GMP batches, but also at bigger scales. Later on, I'll give you more details about the scales that we are covering and the scales that we'll cover later. Okay. Some pictures for you to have an idea about our facilities and so on. So we are based in Spain, in the north of Spain, in the Basque Country, okay, in San Sebastian. And here you can see this is the building where we are. It's a rebuilding where our labs, clean rooms, etc. they are based, as well as the offices. Some more detail about the labs, the R&D lab where we are offering services for the market, such as cloning, manufacture of R&D batches at a small scale, process optimization at a small scale, and so on. But also we are doing our own R&D, whose main aim at the end of the day is being transferred to be offered for services as well. Okay? We do have a quality control department because a great part of the analytics that we are doing to characterize the final product the lentiviral vector, they are made in-house. We have this well-developed and validated method in-house. And this is a clean room where you can see the kind of batches that we are manufacturing for phase 1-2 clinical trials. So this is a clean room where you can see these are 4 reactors in a row. And what we make basically is that we collect harvest coming from these 4, and we process it under a single downstream process. So at the end of the day, what we got is a lentiviral vector that has been purified, concentrated, and buffer exchanged, and that we can dose by using different fill and finish formats. This is where we are now. But next year, so very soon, we are working on that at the moment, we will move to a new building that will be based in the same technology park in San Sebastian, okay? And here we will manufacture additional clean rooms, up to 6 clean rooms, and that's why it will give us greater capacities and much more flexibility and versatility. So this is very important. Here you can see the scales that are available, and some of them that they are coming in the midterm. So the good advantage is that they will cover different intermediate scales, allowing the adaptation to the developmental plans to a given customer in a very customized manner. So the scales will range from 2.4, 4.8, 10 square meters, 30 square meters, 60 square meters to 200 square meters. So you can imagine the kind of, like adaptation that this will provide. A little bit about our process. This is a summarized, slide, but I think it can be useful. So what we make is that, first of all, a master cell vial is defrosted, hectonate 3T. Then cells are expanded during 8 days, firstly in flasks and then in cell factories. And at day 8, the reactor is seeded. It could be different batch sizes. At day 14, transient transfection takes place by using PEI as transfection agent. And later on, harvest is collected during a time that could vary from 3 to 5 days. At the end of the process, downstream takes place. This is obviously a faster process, but not less important. Indeed, we are putting great effort in optimizing also this downstream processing because of the recovery and also because of the purity impact on functionality. So this downstream processing is composed by a clarification step, anion exchange chromatography, sterile filtration, TFF, to buffer exchange and concentrate, and finally fill and finish in different formats such as vials or bags, depending on requirements. So as a summary, we are a GMP CDMO. We are fully focused on lentiviral vectors, and in this case, we are not developing our own pipeline, which means that we are giving full priority to our customers' requirements. There are dedicated upstream and downstream optimization departments. Continued improvements are applied to the services we are offering, and we are also adapting processes to specific lentiviral vectors. And also, well, it's relevant to mention that we have this own R&D department that is working in very ambitious and interesting projects, such as the development of a serverless adherent production process, the generation of a stable cell line, and also, well, I want to mention this worldwide patented technology, Lentisoma, that is a non-integrating lentiviral vector that offers, well, interesting possibilities thinking on safety-related issues. So, well, basically this is all I wanted to present today. Thank you very much for your attention. In the event that you have any kind of requirement related to lentiviral vectors, please do not hesitate to contact us, and we'll be very happy to arrange a meeting and to have a dedicated discussion. So thanks a lot and have a good day. Bye.